My name is Keith Lahr. I'm Associate Professor in the College of Biological Sciences. I'd like to thank everybody for coming out for today's STOR lecture. We've got a real treat today in that we've got one of the discoverers of one of the key kinase complexes within cells here to talk to us. He's one of the best speakers that I've seen. Scientifically, he's top-notch. He's a professor at the Whitehead Institute at MIT. He's done spectacular work, but he's told me to be very short about the introduction, so I'm going to leave it at that and let him go ahead and tell us all about the regulation of mTOR and growth.
Okay. Thank you, Keith. And thank you, everyone, for coming, and thank you for having me. It's actually my first time in Davis and in Sacramento, so it's this area, so it's great to be here. So I'm going to talk about two things, actually. I'm actually going to do what probably all of you who are students have been told not to do, but I'm going to get away with it. And that is try to tell you two stories when I should only be telling you one. And that's because I couldn't really quite decide. So we work in these three areas. One is growth regulation, which is a lot of around mTOR, and I'll talk about that. In cancer metabolism, which I'm actually not going to talk about, although there's been lots of connections now between mTOR and metabolism that we're excited about. But we also have always done a little bit of tool development, usually about sort of trying to develop high throughput tools to study mammalian cells, and we did a lot of imaging and RNAi, and most recently sort of this CRISPR-Cas9 system, which I think all of you are well aware of. And so I'm going to talk about some large-scale screens using that system, which are mostly related to cancer, but not complete. So hopefully I'll get through these things. And so one of the things that we've studied for many, many years, in fact, since I was a graduate student, is the MTOR pathway. This is a major regulator of growth at multiple different levels. When I was in Saul Snyder's lab, we identified this MTOR protein, and when I went on to the Whitehead, I sort of took this to sort of continue working on it. And so I am a bit of an odd example of continuing to work on my graduate thesis project in a way. This was not really completely by choice, but when I went to the Whitehead, I really didn't have any choice of what to do, because it's really the only thing I knew how to do. And so that explains why I've continued working on this system. But it's actually turned out to be quite interesting and connected to many, many different physiological states as well as disease states. And so when I first started talking about mTOR, people would be like, oh, what is that thing? And no one really cared. But now people seem to care much more about this. And the direction that we got to this, and in fact the direction that most people come to it, is from the study of this molecule rapamycin, which I think many of you probably have heard about. This is a small molecule drug made by bacteria on this island, Easter Island. This is where the soil sample was captured. Many different clinical uses shown here. Lots of excitement actually currently in the anti-cancer area, because we're finally starting to define biomarkers that predict rapamycin sensitivity. In fact, this molecule has been thought about as an anti-cancer agent. You could work back almost to the 70s, actually, but only now are we starting to have biomarkers that actually predict a good response. The aspect, though, of this molecule that took it from kind of our world to more the New York Times world is the fact that this prolongs lifespan in many different model organisms, from yeast to mice. This has been reproduced by a number of different labs, and makes it, I would think, probably unique amongst small molecules and having this robust sort of lifespan and elongating effect. This is leading many people to sort of self-medicate themselves, rapamycin. This, I think, is a serious side effect that would prevent me from doing that, but is leading to sort of stories. I think the latest one was in Bloomberg News, sort of arguing, is this sort of the anti-aging molecule, and maybe we don't need to find any more of these. I'm not really on that bandwagon. So we haven't really done much in the aging field, but we have been interested in the pathway that rapamycin targets. And I like to show this very old slide from 2002, because it shows in a more simplistic form what are some of the big picture questions that we've been going after. So rapamycin targets this large kinase called mTOR. In fact, exists in several multi-protein complexes, one called mTORC1, which is what I'll talk about mostly today. It regulates many processes that consume or generate large amounts of energies, for example, autophagy, translation, ribosome biogenesis. And these somehow converge to set the growth rate of the cell, and therefore how quickly it acquires mass and how quickly it reaches a certain size. And this has some, I would say, amorphous connection to cell division, that is the relationship between growth and cell proliferation control. We've actually not done very much down here. What we've been mostly interested in is how does this pathway sense the large variety of signals that it can detect. And I think this is what makes this pathway very unique compared to most other pathways that sense sort of a circumscribed set of signals. This thing will sense all kinds of growth factors, energy levels, all kinds of nutrients, oxygen levels, stress, osmotic stress. Anything you do to the cell, somehow mTOR knows. And it regulates this growth rate. And this tells me the cell cares a lot about regulating the activity of this pathway. And it also sets up, I think, an interesting biological problem, because how do you integrate all of these signals to give a convergent input to this kinase? And to a large extent, that has been the question that we've been trying to pursue. This is a signal integration question, which is common to any signaling pathways, but I think it's particularly challenging in a system that pretty much senses everything the cell can detect. Now, when we think about this pathway at a higher level, at an organismal level, the way we tend to think about it is that this is a system that detects the availability of food in the environment to whether the animal is in an anabolic or a catabolic state. I think this helps explain why the pathway has so many impacts in different physiological systems, because although we don't tend to think about this in our daily lives because we are, if anything, overfed rather than underfed, I think our evolutionary history, the history of most animals out there, and sadly of many human populations, is really a food scarcity, where you have to regulate almost all physiology into whether the animal is in a fed or fasted state, and likely periods of sort of fasting and feeding, while for us are periods of mostly hours in between those, likely these were actually longer periods where there's sort of boom and bust cycles of food or no food. And I think that explains why this pathway is connected to so much physiology, although I will admit that this is a great oversimplification. Oops. So here I'll show you a little bit of, I don't know why it's doing that. Show you a little bit of data on what actually happens to liver size when you perturb this pathway. So for example, here's a control mouse liver, control mTORC1 activity. Here we've genetically inhibited or activated this pathway, and you can see the liver shrinks or it gets bigger. Not dramatic effects, less than 50% changes in size. What's interesting though is if you put these animals in a fasted or fed state, the livers don't change. And that tells us that this is the pathway that's mostly connected to sensing this nutritional state. A mouse, if you starve it for about 48 hours, which is typically all that you're allowed to do from an animal care point of view, will shrink the liver to exactly this. And if you take this liver, it won't shrink any further, and likewise this one. So again, this connects it to this sort of being a nutritional sensor. There's other pathways like the Hippo pathway. They're probably more involved in setting what is the absolute size of the liver. That is, how does the liver know it should be a particular size in a particularly sized organism? This is the one that connects it to the environment. When we think about these liver cells, we sort of compartmentalize the inputs in two simple categories. One are local nutrient levels. This is something we've been particularly interested in. And the other are hormonal cues that are sent out by specialized tissues that tell other tissues that the organism is a fed or fasted state. And of course, insulin is the best known example of this, but there are probably several dozen signals that we can put into this kind of category. And so this pathway needs to integrate these two classes of signals, along with all those other ones that I showed you before, to drive a coherent growth signal. And so our question has been, how does it do this? Now, we know a fair amount about this, and a lot of this is not from our work. A lot of it is through this tumor suppressor called TSC, where you can see all these different pathways that themselves have tumor suppressors in them are integrated at the level of TSC. And TSC is an inhibitor for a small GTP. It's called Reb. Reb is related to RAS. When Reb is bound to GTP, it binds to this mTOR complex 1, mTORC1 complex, and really strongly activates its kinase activity. And in fact, in genetic experiments, we see this protein being as important as the key subunits of mTORC1. Okay. So how exactly all of this integration happens, we don't know, but we know it does go through this pathway. The input, though, that we've been fixated on does not go through this system, and these are amino acids. And amino acids are quite interesting because if you remove them from the media of a cell, this pathway is inhibited. And moreover, none of these inputs work anymore. So somehow it has a trump card, which basically vetoes the power of any of these signals from activating the system, does not go through this. And when we started this work around 2008 or so, this is what we knew. Amino acids regulate this pathway, but we didn't know any of the actual mechanistic insight. And so what I'm going to tell you about really is our efforts to understand this, which are still ongoing, and we don't have the complete answers here. Now, before I do that, though, I want to convince you that studying amino acid sensing is actually interesting. This is something that some people at my institution said, who cares about amino acids? And I often thought that was rather strange because no one says who cares about glucose or who cares about lipids, but amino acids, of which there are many and some are essential and non-essential, are almost like the sort of the poor stepchild of nutrients. A lot of people don't give them much respect, and so I added this slide here. One is that they actually are dynamic. And I think many people in this audience know that depending on feeding and fasting cycles, they go up and down. Muscle is a reservoir of amino acids, and so they do change. Not as much as glucose, but they change in our blood. They promote a lot of interesting physiology, so for example, interesting effects on muscle anabolism, interesting effects on satiety, interesting effects in other organisms like mosquitoes and egg laying. Turns out that they detect amino acids in our blood as the signal to lay eggs. And a lot of this is through the mTORC1 pathway. And so I would argue this is one good reason to study the system. In addition, there's increasing evidence that mild alterations in amino acid metabolism, not the inborn errors of metabolism that many people are aware of, but more mild ones, are associated with diseases like pancreatic cancer or metabolic syndrome. And so I think this is going to be an increasing area. Now, there's a community out there, and we talked about this a little bit at dinner last night. There's a community out there that's always loved amino acids and always connected it to mTOR, and these are the muscle builder people. And in fact, if you go to any of their websites, this is an old one, you go to any of their websites, they're all over mTOR. In fact, they've been all over it way before many of us have been. And they sell products such as A-Bomb, interesting name, that promotes mTOR activation. And here you can see the nutrient arm. Here's the growth factor arm, kind of like I showed you. They put mTOR in the nucleus, which is not right, but otherwise it's not so bad. So this one's one of them. Strange name. This is my favorite, actually. This is a new one. It's called CreamTOR, which I find a rather off-putting name. But what I like about this one is that they actually realized that they had to do mTORC1 activation. You can see the small print here is mTORC1. This is actually the original one. This is the first one. It's actually not on the market anymore. It's called Anator, and you can see it's got interesting ingredients, including my favorite, GeneTOR, as one of the key ingredients. Now, all of these things actually do work. They do turn on mTOR. We've tried them. But they're no better than sort of sigma amino acids. Will do the same thing. And they cost a lot. This thing is like what used to be like $100 or something for this sort of bucket here. Now, the truth is, I'm making a little bit of fun of this, but they do have effects. And in fact, if we could understand how this pathway senses amino acids, we might be able to drug this system for the benefit of patients that do have issues with muscle mass, particularly in the elderly or the inactive population. That remains to be seen, though, if that is going to turn out to be true. So the answer to how this pathway senses amino acids is intimately involved with another set of GTP Aces, which are also related to RAS, but much, much more distantly, and are pretty weird GTP Aces because they're heterodimeric. And these are the RAG GTP Aces. And a student of mine, ex-student of mine, Yasmin Sanjak, a while ago identified these as key mediators of this input. These both combine nucleotides, which make them a real pain to study at the biochemical level, but we know that the nucleotide state of both of them matters in complicated ways that I'm actually not going to discuss today, and that both these proteins, by putting them in the appropriate state, can either signal the presence or the absence of amino acids. And they're absolutely essential for doing this sensing. And this has now been shown in a number of different species to actually be the case. Now, these are MEFs in which we've knocked in the mutation that locks one of these, RAG-A, into this GTP state. These are the control cells, and here's one of the outputs that we use for this pathway, S6 kinase phosphorylation. You can see amino acids nicely activate this. When you have this mutation, though, you can see there's no inhibition of this pathway in the absence of amino acids. In fact, they think amino acids are there even when they're not. This is done in a relatively short time point, but this will persist for hours, in fact, until these cells die from the absence of amino acids. And you can see this is true even though this activated allele is not expressed well, and this is a protein stability issue. For some reason, the activated protein is unstable. It's not a mouse engineering issue. Alejo, who was the postdoc who did these, wanted to get live mice with these mutations. We had all these dreams of sort of having amino acid starvation-resistant mice, of doing longevity studies, all these things, but he never got any animals. They never appeared. He'd find fragments of animals, but he never found an intact mouse. And so what he realized was happening is that these animals were being born and were dying perinatally, and the mothers were eating them. And so we never actually found them. And so he started delivering them by cesarean, and in fact, by cesarean, at term, they look perfectly normal. Here are the double mutants, these are the hets, these are the wild types. They're a little bit smaller, but otherwise, they're normal. And so what he did is he put them into a humidified cage and basically did a time to starvation type of experiment, which has been done previously by others in mice. And you actually get fairly stereotypical effects when you do this with wild type mice. And that is that animals typically perish in about 24 hours or so. Okay? These animals die in about half that period of time, the double mutant. And I think some of you will recognize that this phenotype is very similar to what happens if you inhibit genetically autophagy. And the argument has been that when a mouse is born, it has very little levels of fat tissue, and it basically has to break down its own components to generate nutrients and energy to survive this perinatal starvation period. And this, of course, makes a lot of sense from an evolutionary point of view, because if a baby is born and immediately needs to get fit, this would not be selected for. Again, not humans' typical experience where babies are fed pretty quickly, but certainly out in the wild you can imagine how this would matter. And it turns out that this GTP Ace is key for this response. I'm not going to show you any of the data on this because it's been published, but what's happening here is that these animals cannot trigger autophagy. They cannot therefore maintain their glucose levels, and these animals basically die when they go into a hypoglycemic crisis, is when they perish. And we can fully rescue this by giving amino acids that they can use for making glucose in their liver, by giving gluconeogenic amino acids. And this led us to realize that this pathway is not only sensing amino acids, but also glucose. I'm going to tell you today about amino acid sensing, where we don't know too much, but we don't know anything about how this system actually senses glucose at all. But we think a lot of the similar players are involved. Now, one of the things that he thought about was, well, if we delete one of these and replace it with a null, maybe we'll get mice that will survive longer. And here are MEFs that I just showed you. You can see very little protein, fully resistant signaling to amino acid starvation. You can see if we delete one copy, half the amount of protein as you would expect. Also fully resistant, at least in these MEFs. Now, that might not be true in some tissue in vivo. And indeed, these animals now are born. They're not born at Mendelian ratios, but we actually do get some of them. Here's one of them here. Here's another one. They're very easy to genotype because they're a little bit grayer in their hair. This is not a sort of pro-aging phenotype, which is what we originally thought. They seem to have some melanogenesis issue, that their hair becomes sort of prematurely gray, but we don't understand that yet. Now, these animals do die eventually. They die a couple of months into their life, and they seem to die of seizures. And yeah, there's actually an interesting connection, because there's a human gene that also regulates the state of GTP loading, the RAGA, in humans that get epilepsy. So there's some connection between this, which maybe if I remember, I'll point out afterwards. So we haven't done too much with these animals. One thing we've done is some activity measurements where we put them in metabolic cages in both a dark and light cycle, and we put in fed or fasted animals. And you can see the mutants are certainly more active in this dark period when mice normally feed, particularly when they're fasted. They're much, much more active. And this and other experiments led us to conclude that these animals can detect that they're hungry, but they can adapt their metabolism to this fasted state. And indeed, these animals frequently seize and die during this time, especially if they're touched. They seem to have a seizure. And so they may be having what's been called a hypoglycemic seizure, although I don't really get what that is. So we're just starting to study these because we barely have any animals so far. One of the things we've started to do, though, is to knock in this mutation using the same approach of the mutant over null into different tissues. Here's a wild-type liver, fasted, rapidly fed. I think this is a 40-minute refeeding. This is an output for mTORC1. Very strong activation of the liver. It's classic sort of feeding-induced mTORC1 activation. If you take the mutants, though, fasting does nothing. It does not turn off this pathway. And this is quite interesting and goes against what I told you before, where I said, look, you need both this growth factor and this nutrient input. Here, all we've done is perturb this nutrient input, and the liver no longer senses the fasted state, at least vis-a-vis mTORC1. This, I think, in retrospect, makes quite a bit of sense because the liver gets this massive bolus of nutrients coming from the GI tract. In fact, it sees levels of nutrients that are many-fold higher than any other tissue. So it might be specialized, at least for this system, to just look at nutrients, sort of be a nutrient sensing hub for the rest of the body. But that remains to be seen if that's true. Nevertheless, this experiment is certainly true. The 1 point mutation in what we thought was just a nutrient sensing arm eliminates the response to fasting. So these proteins are necessary and sufficient to mediate this amino acid signal, but the real question became, how do they actually work at the mechanistic level? Because the obvious answer that they're sort of regulators of mTORC1 kinase activity turned out not to be the case. And in many kinds of experiments, including one from ex-postdoc Roberto Zonco, who I understand was here in Davis recently, this is a movie that he captured. What you can see is when you add amino acids, which is this little white box it shows, and you look at mTORC1, it rapidly relocalizes to these little puncta inside of the cell. And what these turn out to be are lysosomes and late endosomes. And what's happening is mTORC1 is moving to that compartment. And if you take these cells that have this activating mutation mTORC1 is always in that compartment. So there was a correlation between this localization and the activation of this pathway. And I want to stress, it's not going inside the lysosomes. It's not just on the surface of the lysosome. This led to this kind of model where when there's no amino acids, mTORC1 is somewhere. We don't know what that is. And when you add amino acids, it docks on these RAG GTP Aces, which we mostly think is a docking station for mTOR1. And that allows it to come into contact with Reb that we and others had shown was on the lysosomal surface. At least some portion was on it. And there's other regulators, for example, this GEF and scaffolding protein that has this unfortunate name of regulator is also found on this compartment. None of these are transmembrane proteins. This one has a little lipid tail that anchors it to the surface of the lysosome. So this is just a coincidence detector. Amino acids regulate this protein to allow mTORC1 to dock, and then all those other signals that I showed you regulate this GTP Ace and allow it to activate mTORC1. So you need both signals for this system to be turned on, and that nicely explains why when there's no amino acids, it doesn't matter what you do here, because mTORC1 is just not in the right place to be turned on. And we have done many experiments that I think argue that this model is largely correct, although certainly there's going to be details that are perhaps not correct. And I think that this has been supported by a number of different groups. Brendan Manning has really nice data showing that TSC, that inhibitor of Reb, comes on and off the lysosome in response to insulin signaling. So I think connecting that branch here as well. Now one of the things that we were particularly interested in is understanding what it is about lysosomes that this pathway might be sensing. And so one attractive idea was that actually it was the contents of amino acids inside the lysosomes that would be sensed. And if that were to be the case, you need some way of getting that signal outside. And this is what I'll tell you about afterwards. And so the model that we eventually came to was that amino acids got into the lysosome. Here we're showing them as going through transporters, which we know exist. But the other way could be by simply degrading proteins or peptides. And I think there's a sort of a bit of a ground shift going on right now as to where amino acids actually come from. Are they mostly from proteins? Are they mostly from free amino acids? I think there's going to be quite a bit of changes of how we see that inside of our own bodies. And so we think they get in to the lysosome, and then through a pathway that involves this vacuolar ATPase, which Roberto was working on, which I won't discuss too much, and this series of complexes, this protein gets loaded appropriately, mTORC1 docks, and the system's turned on, as I told you before. We now think this is the on switch for this pathway. And more recently, we've discovered a series of complexes, which go by these names, Gator, which seem to be the off switch. And so we think there's a distinct on and off switch. And in fact, we think the off switch starts in the cytosol, while the on switch starts inside the lysosome. This protein is an inhibitor of this guy here, of this RAGA. And in fact, this is the complex. There's 3 proteins in here, which are now thought to be the most common cause of familial epilepsy, that is loss of function mutations in this complex, which fits quite nicely with the fact that when we put this one in the activated state, which is what you'd expect when you delete this, our mice seem to have seizures. And so there seems to be perhaps a connection there between our data and the human data that's the clinical human data that's out there. And I'll get back to this a little bit afterwards. So at this point in the story, I have to say we felt pretty happy. We never imagined there were so many proteins here. These are all protein complexes, and so there's about 20 proteins in here. So the cell devotes a lot of energy to regulate this. And so we thought, okay, we have a lot of these pieces, and so that's why I put this kind of recent graduate type picture. This is not someone I know, I should say. Happy. But then we started addressing the question that probably many of you are thinking about, everyone always asks me, is what are the sensors? And it turns out that none of those proteins are the sensors, as far as we can tell. This has turned out to be a much, much harder problem than we ever imagined. In fact, so now we feel like that, sort of a little crying baby. And the reason is because there's a lot of amino acids, and we didn't realize how many mattered. They come at very different concentrations in different tissues, and they can be in different compartments. And I already told you that there's probably an on and an off switch for these guys. And so all of a sudden, what we had originally thought, like when Roberto was in my lab, we used to talk about the amino acid sensor, the amino acid sensor. It's pretty clear that that is completely wrong now, and that there are many of them. So thus, crying. So the first question you have to ask is, are there many sensors, right? And one way of doing this is by challenging the system with individual amino acids. And if you put any single amino acid, it will not activate this system. You can put any 2, and it won't activate this. Once you get to 3, though, and in particular this combination, leucine, arginine, and lysine, which we affectionately call LRK, this is sufficient to activate this system. So no amino acids, all 20, it turns on. All combination of 2, LR is not bad, but when you put LRK, you reconstitute this activation. Okay? It's not that other amino acids have no effects. They do. They're minor. But this combination in our hands, which we've tried all possible combinations, including combinations of 4, is the best input. We know that these are not substituting for each other. Particularly, you might imagine arginine and lysine, which are related. They will not substitute for each other. They are not necessary for each other to get into different compartments. And so we're very certain in saying that there's at least 3 binding sites here. Okay? And if there's an on and off switch, I would say there's probably 6 binding sites that we have to care about. That's assuming there's no redundancy for each of these. Okay, so if you're going to go call something a sensor, which is what everyone's been looking for in this field, I think it's important to establish what would make you say you believe it's a sensor or not. So one is pretty obvious. It should bind amino acids at the affinities at which they're sensed. The big caveat for us here is that amino acids are very high concentration, and there's some evidence that in lysosomes and vacuoles, they can actually be at tens of millimolar concentrations. So we don't know exactly what they are, but even if they are, these concentrations are not really compatible with doing traditional biochemical assays here. So this is going to be a problem for us. There could be many, but there's probably should be required for sensing. Yeah, there could be redundancy. We could probably deal with that. You should be able to make a mutant that makes this pathway insensitive to amino acids. You might need some structural information. And then lastly, the thing that would really convince me is if I could make a mutation and I would change the specificity from one amino acid to another. That's what would really convince me that I had a sensor. There's lots and lots of caveats in actually doing this experiment. So I'm kind of putting this up as a potential report card for what I'm going to show you afterwards. Sorry, it's quite a bit drier here than it is in Boston, I think. Is that a true thing? Yes. What? Yes. It's really dry. I guess I should say it's really humid there. Okay, so if you've been following what I'm saying, we need a transmembrane protein at the lysosome. And in fact, I already told you about this VATPase. And Roberto is pursuing the idea that this might be involved in amino acid sensing. And I'm glad he took this with him because this is not a fun thing to work with. It's got 16 subunits and it's never been reconstituted in a recombinant way. But he has some nice evidence suggesting that might be involved. We, though, started working on this other protein, SLC38A9, which you see has multiple transmembrane domains. And we had identified because it bound those GTP Aces as well as that GEF regular. We actually identified this many, many years ago, but have had a very hard time working on it. And when we started working on it, there was nothing had been done on this. So there was no data on this protein whatsoever. So it had never been studied. This has 11 transmembrane domains. It binds to these guys, and it binds in an amino acid sensitive way, which was a nice thing to see. It has this unique N-terminus, and that's in fact the thing that binds these guys. And this N-terminus is only found in homologs of this protein. Okay? And this N-terminus is necessary and sufficient for binding the RAGs. And you can see this is towards the cytoplasmic side. What really captured our attention is that this is part of a sodium-coupled family of amino acid transporters. And this was, in fact, the first protein that we ever identified that had any connection to amino acids whatsoever. All those other proteins I talked about have really no homology to anything, so they don't really tell us much. But I want to stress that this protein had never been studied, and that whether it couples to sodium or whether it transports amino acid, I would say, was largely unknown. And this is expressed at very low levels, and there's splice variants, including some interesting ones that delete this N-terminus. I'm not sure what those would be about. So most of these SLC38 family members are actually not at the lysosome, they're at the plasma membrane. And so this was a problem for us, but it turns out that this one is completely at the lysosome. This is a lysosomal marker. And in fact, it does not change minus plus amino acids. And like many lysosomal proteins, it's heavily glycosylated, presumably to protect it from the nasty environment inside the lysosome. One of the things that really captured our attention is when we overexpress this protein. Here are a couple of controls that we overexpress, and this is the kind of signaling I showed you before, minus plus amino acids. There's no perturbation. But when you overexpress this one, we largely rescue this minus amino acid state. And indeed, this is what you might expect when you find a receptor, right? So if you overexpress the EGF receptor, you activate the MAP kinase pathway. It's very typical for sort of apex members of a pathway that they tend to be limiting, and that's what seemed to be happening here. And moreover, if we made a point mutation that eliminates capacity to bind the RAGs, this didn't happen. So it's clearly through that binding activity. Indeed, we can just take that N-terminus and overexpress it by itself and get this activation, suggesting that N-terminus acquires some kind of active conformation when it's cut off from the transmembrane domains. And what I forgot to mention, I always forget to mention this, is that in other transporters, it's the transmembrane domains that bind amino acids, suggesting that there might be some conformational change there that's led to to the N-terminal, changes in the N-terminus. So does it actually transport amino acids? What's the evidence that this thing will bind amino acids? And this turned out to be exceedingly difficult to do. It was very clear early on that if it does transport amino acids, it does it really, really badly, which is what it says here. But we finally did load it into proteoliposomes and did sort of hot amino acid transport assays, and we found that it transported arginine, but really badly, with a Km around almost 40 millimolar. This is actually not that bad. It turns out that other lysosomal transporters have Kms in the millimolar range. For example, PQLC2, which is an established arginine transporter of the lysosome, has a Km around 5 to 10 millimolar, and this supports the idea that transporters at amino acid are high concentration. The other reason is they were probably driving this in the opposite direction because of the way it orients in these liposomes. We're actually driving it in the direction we think is backwards. But this led to the question is, could this have a role in sensing arginine? Remember, that's one of the amino acids I told you mattered in this system. And so we knocked it out using the CRISPR system, and these are true knockouts. In fact, we've done rescue type experiments with the cDNA, and it fully rescues them. Here you're looking at leucine at different concentrations. There is a small defect in how it senses leucine, but not a big one. It can sense leucine. In contrast, it really can't sense arginine. And again, this is fully rescuable by putting in the cDNA, which now lets us do a lot of the structure function stuff that we can't really do based on looking at its sequence. We can't really predict a lot. I want to emphasize that this is part of the on switch. It can still turn off when you remove arginine, but it doesn't seem to be able to sense the readdition of arginine, which again supports the idea that the on and off switches are different. So if you go through what I showed you as sort of a report card, I would say we don't know whether this is true because we don't know the concentration in lysosomes. We're trying to figure that out. It is required for sensing. We can make a mutant. I didn't show you this. And this we have no idea. And so when I've talked about this before, I've kind of put this in the B range. It's definitely not an A grade, but it's sort of a B range. But I'm pretty confident in saying that ultimately we're going to prove that this is actually a sensor because it's certainly in the right place and it looks like the right thing and it's bound to all the right proteins. And so if it's not a sensor, it's an incredibly cruel joke that's being played on us. But I'll be the first to admit that we have not proven that it's a sensor. And so we've been sort of shocked by the level of complexity. I thought there would be some kinase that senses amino acids, phosphorylates, mTOR, and that'd be the end of it. But we have this on switch, we have this off switch, we have this putative arginine sensor. And moreover, it's very clear that there are multiple amino acid inputs coming in. So for example, we know there's an amino acid input coming from here. And we know there's another amino acid input coming. I didn't talk about RAG-C, but this is a gap. It's an inhibitor of RAG-C. There's another amino acid input that comes into this. And so there's at least 3 places where there's amino acids feeding into this, and perhaps more of them. And so there's a lot more for us to figure out, which is good. So we sort of continue having a job, I guess, and continue getting grants. Okay. Now, I've always been interested in tool development. I'm going to switch gears here. For mammalian systems, partly because, or I should say mostly because I've been very jealous of the power of genetics in yeast. And I've worked at an institute where we have Jerry Fink, really one of the pioneers of yeast genetics, and we've not been able to do any of that kind of stuff. I think we really are at the cusp of being able to do that now with human cells, mammalian cells, in culture, not organisms, human cells, because of this CRISPR-Cas9 system. I really think that we're going to look back at this time as an inflection point in our study of these cells, again, in culture. And I'm not going to go into this details a lot on this system. I think all of you know that this is a nuclease that under the influence of a small guide RNA will go hone to a particular place in a genome, cut it. When you get imperfect DNA repair, in our hands and the cells that we use, you very frequently get a null allele in that situation. And we didn't discover any of this, and we didn't develop any of these systems. What we have been doing is taking this to a high throughput level and trying to exploit it, make it into a true unbiased genetic system. And the work I'm going to tell you about is through an amazing student, Tim Wang, who's a joint student with my lab and Eric Lander's lab, who's mostly at the Broad. And so what we can do with this system is make large-scale libraries. We can make lentiviruses that encode Cas9 in them. We use what's called a OneVector System, and the guides that we care about. Again, large scale, we make these on chips. We make large scale libraries. In fact, the first library we made was targeting 8,000 genes with 10 guides per gene, so 80,000 guides. And the way we do the experiments is that we take this library, we infect cells, so each cell gets one guide, and then we wait. over time. We take a sample of genomic DNA at the beginning, and then we wait usually a couple of weeks. The cells have divided usually 15 times. And through deep sequencing, we compare the abundances of those guides at time 0 versus that later time point. And the inference that we make is that if the abundance of a guide goes down, it was targeting a gene, knocking out a gene that the cell cared about. Right? So it reduced the fitness of that cell. If the abundance of the guide goes up, the cell liked losing that gene, and the fitness of the cell went up. Okay? So it's a cell number based screen, which is what lots of yeast geneticists have done for a long period of time, where we don't really know what's happening in terms of proliferation, death, growth. It's just cell number, so you could call it just fitness at the end of the day. And we've done this across many different genotypes that represents sort of heterogeneity of human cancer cells. We've also done it in the presence of drugs or viruses or things like this, and looking sort of before and after that treatment. Those tend to be positive selection screens, which tend to be much easier, and I'll show you just one example of that at the end. So the first screens that we did worked really well, but it was very clear that not all the guides were working. And so we did quite a bit of analysis, tiling large number of genes we knew were essential, figuring out rules that we could apply bioinformatically to make better libraries. And the library we use now is the 2.0 library. I think this library is about 90 to 95% of the best that we could get in this system, but it's very close to that. This targets 18,000 genes. The reason it's not 20,000 is because we didn't target the olfactory receptors, partly because they're so homologous that it's hard to make a specific guide to an olfactory receptor, partly because we didn't think they mattered, but I think that's a bad reason for that. So there's 180,000 guides to this library. So what I'm going to show you is our application of this library, and I'm not going to go deep into biology at all. I'm really trying to sort of advertise this system because I think it's really cool. I'm going to show you our attempts to define the essential genes of human cells using this, attempts to define the oncogenic drivers of cancers, to look at synthetic lethality interactions in cancer, and there was one, and using this in correlation type analysis to define the members of complexes. You'll see, sort of try to like infer biochemistry based on this. I think that application is pretty cool. So the way we look at the data is we make a score for each gene, and then we rank it from most negative to most positive. So these are all essential genes. These are genes that help the cells. This is heavily enriched for tumor suppressors. In fact, you discover the entire list of tumor suppressors if you look in this list. You can see it's asymmetric. More genes are needed for the cell to be happy than are sort of suppressing the fitness of these cells. The number one hit here is always B10. If the cell hasn't lost B10, that's the number one gene at that point. Now, if you define some cutoff here, you get about something like 1,600 genes that you could define as essential. All the things you expect to be there, ribosomal, RNA polymerase, DNA, are all there. The ones that are not are the ones that have paralogs. So there's redundancy in those. There's actually some other interesting things in there that we're not really following up. So 14% of these are uncharacterized. So there's no yeast homolog, and there's no annotated function to these. These are the genes that typically look like C11 or 59. So they're just a chromosomal localization. So Tim tried to see, could there be something that united some of these genes? And he looked in lots of different data sets. And what he found in a proteomic data set is that many of these seem to be nucleolar, so nucleoli proteins. That is, they're localized to the nuclei. Someone had sort of purified the nucleoli and done proteomics, highly enriched statistically versus other compartments that we've looked at. We're not following this up too much. He has sort of looked into a couple of them. Here's one of them. It's clearly nucleolar. This is another one. It clearly interacts with different RNA processing and RNA helicases proteins. This actually might make sense because the ribosome, of course, has diverged between yeast and us, and so you might imagine that you would have to acquire some new functions in the nucleolus to sort of enable ribosomal biogenesis. I think this is a cool area to look into, but Tim is mostly sort of a large-scale computational guy, so he doesn't care too much about this. But I've shown this to other people, and they're like, oh, that's kind of cool, but we're not doing much here. So you can also look at some more broad properties, and a lot of these are not so surprising. So for example, the genes that have very negative scores, that is the most essential, they're very conserved, right? So lots of species have those. Makes sense. They tend to have very few SNPs between human and mice, so again, makes sense. They tend to be less tissue specific, so they're not going to be a liver specific gene. The only one that's a little bit surprising and has been debated in the yeast world is that they tend to also be better expressed. So expression is a pretty good predictor of essentiality. In fact, if you take our data sets and use them to predict yeast essentiality data, where there's pretty good data out there, and you look at one-to-one homologs between the two species, expression data is better at predicting that than RNAi screening data is. It's nowhere near as good as CRISPR-based data. We can actually very well predict those. But expression is actually pretty good as an indicator. Whether this is true in many other systems, we'll have to see. But certainly in human cells and culture, it's pretty good at predicting essentiality. Now, the area that one really cares about is more cancer, I would say, or at least we do. And I'm going to show you a couple of examples that we've done in cancer systems. And the first one we did was in CML versus Burkitt's lymphoma. These have BCR-ABL translocations. These have overexpression of MYC by a translocation. And so if you do these screens and you get these scores for all the genes, and simply you do clustering, what you find is that these two cluster together and these two cluster together, which kind of makes sense. So that is, the dependencies of these two classes of cells are most similar to when they come from a similar type of cancer than not. So you would expect this. Now, the truth is there are very, very few genes that are driving this, because the vast majority of genes that are essential in any particular cell line are the same. Right? They all have the ribosome, they all MTOR, all that stuff is exactly the same. Now, you can define a couple of different classes of essential genes. Some are the core ones, which I told you about before. These are all heavily negative scores in all lines. There's an interesting subset that's very rare, which we call the cell line specific set. You can see it's only essential in this one cell line, so it doesn't correlate with the type of cancer it is. These almost all fall into one category, which is the following. Here's an example, DDX3Y. This is a helicase on the Y chromosome. And what you can see is that this one has a paralog on the X chromosome, and for some reason this one's damaged in this one cell line. So together these guys encode an essential function, but in one cell it's lost the paralog, and therefore the other one becomes essential. So you could imagine some hyper-personalized medicine approach where you would target this one for this cell, but that would obviously be rather silly. Incidentally, this is the only gene on the sorry, I'm blanking on the, what's the part of the Y chromosome that's similar to the X chromosome? What is that called? The whatever. Anyways, this is the only gene we've ever found on the Y chromosome that's essential, which our director who studies the Y chromosome was very unhappy about. But so one cell line, one gene is essential. Now, the one that people really care about more from a cancer point of view are these, which they're essential in one type of cancer, but not in others. And the way we get at these is we simply subtract these two scores, the average of these two scores. And that's what's shown here, is this differential essentiality. So these are all essential in CML, these are in Burkitt's. And you get a lot of the genes you expect. For example, ABL is the number one here. BCR is not as good because not all its exons are in the translocation, so it lowers its score. Here you get a whole bunch of different B-cell state-specific factors. You don't get MYC, because MYC is essential in all cells. So it's an essential gene. Indeed, a lot of these transcription factors are seen to be driven by what people like Rick Young and Jay Bradner have called super enhancers. I'm not sure if you guys have seen some of these. There's actually a very good correlation between these transcription factors in all of these different cells. I'm just showing you for Burkitt's here. But in all of these, there's some of them as well here, seem to correlate with super enhancers in these cells. So we did larger experiments with AML, pretty common form of leukemia, lots of different mutations. So you can't simply go from genome data and predict essentiality. And so the question was, can we get the drivers in different classes of AMLs? The first screen we did was in 6 lines, which are shown here. And for each of these, we get the most differentially essential genes, which are shown up here. And I'll tell you right now, in all cases that we've done this, it's always that number one gene that's the oncogenic driver for cancer. It's the one that's most differentially essential. So here's the first one, gives JAK2, and this has a JAK2 mutation, classic mutation, so it makes a lot of sense. This one was a little bit different. PDGF receptor is what's scored. This is not classic. Tim looked into this a little bit more. He did what we call the guide level data. I'm showing you here the averages of all the guides. He went into the guides themselves and looked at them. And what he realized is only some of the guides were scoring. And what he figured out is that there was a translocation of this gene. And indeed, if you look at it, this is the part that's in the translocation. These are the different exons. These are different guides up here, sense and antisense. Only these guides are scoring. And so one of the things that we're realizing is that by having a library that spans 10 guides, so multiple different exons, we actually have an exon-targeted library, not a gene-targeted library. And for translocations in cancer, this is turning out to be incredibly useful. It's very easily mapping the translocations for us, which if you were to hone this down just a few guides, and we've done downsampling, we know we could just go to 3 guides and capture the vast majority of the data, we would lose this kind of translocation data. These guys all have mutations in FLT3, and you can see for 3 of them, FLT3 is very negative. So this is a tyrosine kinase that's mutated in AML, but you can see one of them is not. Okay, and this was weird. It had the mutation, and yet it did not seem to be an essential gene in that cell, and we verified this was no technical issue. And so we said, who is up there on this? And this turned out to be Keras. Okay? Now, the reason this had been missed is that this is not a typical Keras mutation. This is an atypical one, which we did analysis to show that actually is being selected for in cancer and is transforming. But that data immediately told us that it was not FLT3, but it was actually Keras that was driving this cell. And indeed, there's another gene that I'll tell you about in a second, RAF1, that also was scoring. This turns out to be the most synthetically lethal gene with Keras in all the experiments that we have done. And that's shown here. So now we're looking at, I think, at 14 different AMLs that have gone through this kind of pipeline, either that have no mutations in RAS, so they're wild type for RAS. They obviously have other mutations, but not in RAS. Or an NRAS mutation, which is common in AML, or a KRAS mutation. And you can see if you have a KRAS mutation, KRAS matters. They have a negative score, not the other ones. Makes sense. If you have an NRAS mutation, NRAS matters, not the other ones. So the gene that is most synthetically lethal with either NRAS or KRAS is this one, RAF1. And there's an outlier. You can see that one cell. It does not have a RAS mutation, yet RAF1 is essential. And we knew the data was tight enough that this was telling us something here. And so Tim went in and looked at this gene. And it turned out, again, if he goes to the guide level data, this guy is translocated in this cell line. So this is a cell line that bypasses the requirement for RAS by directly activating RAF1. In fact, it just has the kinase domain in this translocation. Now, I have to say I wasn't that interested in this because I assumed that this was BRAF, but this is not actually BRAF. This turns out to be CRAF, which is not nearly as widely studied, and in fact, there's no specific inhibitor to CRAF. And the data that we have would argue that a specific inhibitor of CRAF would be quite nice for targeting RAS-dependent cancers. And I was presenting this to some pharma guys trying to get some money to do some screens. And he looked like his eyes all open. I was like, what's up? He's like, David, that's really interesting. I was like, why? It's BRAF. He's like, no, that's not BRAF. This is actually CRAF in these cells. And so we're now sort of collaborating with a chemist over at Dana-Farber to try to see if we can make some CRAF-specific inhibitors. It may not be easy to do chemically. Okay, so people always ask about tumor suppressors. Here's p53 and its 2 negative regulators, MDM2 and MDM4. And you can see that some cells have wild-type 53, some have mutant 53. And if you look at the score for p53, note this is now a positive score. Okay, this is helping these cells. If you have wild-type 53 and you knock it out, it helps them. So it behaves like a tumor suppressor should. And very gratifyingly, if you look at the inhibitors of p53, they have exactly the opposite behavior. They give negative scores only if there's wild-type 53, not if there's mutant 53. And so this started to make us think that these correlations between the scores of genes, here I'm showing you only 6, but what if you did it for many, many more cell lines? Could those correlations start to tell you something about the relationships between genes? Okay? And I think this is certainly true, particularly when we're going to have hundreds of cell lines. But right now we don't. We only have like 14. So I'll show you the data for 14 of these. Here's for MDM2 and MDM4. Each of these columns here represents a particular cell line, and we've plotted it. These are all the cells that have mutant 53. You can see neither p53 nor MDM2 are score here because it's already mutant, doesn't matter. But then there's this beautiful anti-correlation between these two, which is very consistent with them being antagonists of each other. Here's a cyclin that we didn't know who its partner was. It came up in one of those screens. It's pretty clear it has to be CDK6 because of this correlation. These are highly statistically significant type of scores. Then we started getting more bold and just started taking random genes that we didn't know anything about and saying, what's a random gene that has a correlation with some other random gene? And here's one of them, this thing, LDB1, and that thing. Don't know anything about it. It's got a beautiful correlation here. Okay? And so then you just PubMed it, and it turns out that these are interacting proteins and forms a complex. And so this data would easily predict this. These have correlations in sort of the 0.9 something type of factor. Then we started getting more bold and saying, well, what if we start taking components of a complex? Can we predict the other components of a complex? So we took one of the components of the Fanconi anemia complex, FANC-F. Okay? And again, each of these columns represents a cell line. And now you're looking at the scores for all of, these are the genes that most closely correlated, okay, unbiased. You can see they nicely cluster together. Some cells don't care at all about this complex. Some cells care a lot about this complex. You can see in this list, it was pretty clear that it was making sense, except for this one here. This one was not annotated as a FANC complex. So we were really excited. We thought we discovered a new component of this complex, and literally 2 weeks later, a paper came out saying that this was a component of this complex. So we got a little bit scooped. But anyways, it validated the approach. This is complex 1 of the mitochondria. This is one component of complex 1. Again, some cells don't care about complex 1, consistent with them being more glycolytic rather than oxidative type cells. But you can see these sort of beautiful things that come out. And again, these are all components of complex 1 or assembly factors for complex 1. So again, it pulls out the entire bag. These are not cherry-picked. This is literally the list just down from that. In fact, this complex mTOR2 that I didn't talk about at all is completely predicted by this data set. This is a complex that we purified many years ago the hard way. So I'm a really big fan of this for deciphering these kinds of interactions. And I think human cells are actually really good for this because these cancer cells carry so many different mutations. It's kind of like doing a random mutagenesis and then doing these experiments. And so in yeast, while you might introduce different mutations or you might put it in different environments, here we're starting with this natural heterogeneity these cancer cells come from. And so we'd really like to do this in many, many large numbers of cells to get at these kinds of interactions. And lastly, I just want to show you the robustness of this approach. There have been many RNAi screens that were done to identify host factors for HIV, that is human genes that you need for HIV to infect and kill these cells. These were quite famous screens because they were reported in high-profile journals, but people did meta-analyses between these hundreds of genes, and they actually overlapped less than by chance, which was like kind of, in my view, the death knell for sort of RNAi screens. And so we said, well, let's do exactly the same screen using the CRISPR system. So we got the right cell line, we got the right HIV strain, infected them. We did this in collaboration with Bruce Walker and Nir Cohen at MGH, and we said, what human genes give you resistance to HIV? Okay, that was a simple question. And so here are the data. These are all 18,000 genes that stretch that way. Okay, but you can see there's only 2. Okay, in this experiment. These are positive scores. It's a positive screen, and I think all of you can guess what those genes must be. Human genetics had already told us what they were, and that is the receptor CD4 and the co-receptor CXCR4. There's no other gene that gives resistance. We repeated this screen with a virus that instead of using CXCR4 as the co-receptor, used CCR5. So it's a CCR5 tropic virus. And you do that screen, same thing, and now you get 4 genes. You get CD4, CCR5, and then 2 other genes. And those 2 other genes we figured out are modifiers of CCR5. They're important for getting CCR5 to the surface. And so we haven't learned any new biology, which is kind of sad from these screens, but I think it strongly validates this as being a very robust approach for interrogating this type of biology in a culture system. So we're big fans of this. And so let me finish up. The first part I told you about, the amino acid sensing pathway. Many people have worked on this. I mentioned Roberto's work. Yasmin was the one who first found the RAGs, and Liron, Lynn, and Rachel have done a lot of identifying other components of this pathway. Alejo made the mice that I mentioned to you. And then Z and Shuyu, who are 2 MD PhD students, they did this putative arginine sensor that I talked about. And then lastly, Tim is sort of the heroic graduate student who did all the CRISPR-based work that I told you about. He's assembled a bit of a little team of undergraduates that work with him, but he's really been the driver of this. And so thank you very much, and I'm happy to take questions if you guys have any. So.
We realize that we're kind of at time, so I understand if people have to go, but we do want to have some questions. So Gina, why don't you start us off?
David, that was really cool that the mitochondria complex 1 had PDE12 been identified before.
I think it has, yeah. To some extent, its name is not correct. I forget what it is exactly, but I think it's an assembly factor. It has that name, but it's been connected. And in fact, we had, to be fair to RNAi, we actually had pulled it out on an RNAi screen also before. Yeah, I don't think we discovered anything new there. It has that name, which is historical, but I think it's been connected.
But the fact that it's coming up with that, with your screen, means that it's part of the complex 1 complex in some way. And so I'm not sure.
Or intimately tied to assembling that complex, right? I mean, for example, I mentioned mTOR2, and you look at the data for mTOR2 and you just list it, the first 3 hits are all the components, and then a little bit down are the pathway downstream. And so actually the data is quantitative enough, and I don't know for that guy. You're right, scored pretty high, so maybe it's associated, but we haven't checked. Yeah.
Go ahead.
I'm curious, very beautiful work. When you have charged molecules across a membrane, we start thinking electrophysiologically. Is there any evidence for that at all, like pH differences or ion trapping or anything like that?
Yeah, I think that's a really good question. It's something we've definitely thought about. So we don't see any pH changes in the lysosome with amino acid starvation and stimulation. Now, there might be very small changes that we are missing. But whether there's an electrical change that we miss, we don't know. People have started now to develop methods for patching, electrophysiologically measuring lysosomal activity. And I'm pretty sure I've talked to some of them who've looked at amino acid starvation. I don't think they've seen that either.
But I think you could probably just test it with DNL amino acids, I would think.
Well, the problem is with D amino acids is they don't really cross the plasma membrane. So it turns out that lysosomal transporters typically don't care about whether it's a D or L amino acid. And the reason for that is because I think the cell has outsourced that checking to the plasma membrane. So plasma membrane transporters typically do care, but lysosomal ones typically don't. So I think the assumption is if you got into the cell, you're kosher here, right? But not if you didn't.
Great talk. I was interested in the mouse embryo fibroblasts from the GTP mouse. So you said when they are starved amino acids, you have SXK phosphorylation, but eventually they'll die off. Does that have something to do with, I guess they're not getting food, but is AKT not upregulated as well during that? That kind of...
In those systems, AKT is actually quite suppressed because of the feedback between mTOR and the insulin pathway. I think they die of a crisis of not having amino acids, right? There's only so much recycling they can do over time, but I think they typically die in like the 12 to 15 hour range. And that's been reported before, and I think to some extent it's expected. So.
I may be revealing my ignorance, but why have we seen arginine and lysine? Is that tissue specific? Or?
Yeah. So the question is why leucine, arginine, and lysine? Yeah, I think that's a big question, right? I think leucine you can kind of justify as an essential amino acid. It's one of the most common amino acids in proteins. You kind of can hand wave that. Arginine is a little harder, right, because it's not essential. I think one way, so in plants, I shouldn't say this considering I'm at a place with a lot of agriculture. I think in plants it's been argued that arginine and some derivatives like cannabinine are actually really good nitrogen stores. It's the most nitrogen-rich amino acid, so you might say what's happened here is that this is more a nitrogen source rather than an amino acid per se. Lysine's essential also. It's a big product actually for cattle feed. I've learned recently that apparently billions of dollars of lysine are sold a year. So I don't know. I think it's a really interesting question. Why did nature pick some amino acids and not others? I don't even know why you need multiple ones, right? Because there's no real food sources. You never run into a pile of leucine, right? You just eat leucine, right? So you can do that now, but certainly not in nature. So I don't know.
Regarding methods to look at mTOR activation, I noticed you had a lot of Westerns in like the P70S6K1, phosphorylated the screening 3D9 site. Are there more sensitive methods or other things that you've come across or used to look at?
So are there better ways of looking at mTOR1 activation than S6 kinase phosphorylation? Is that the question?
Have you had come across more sensitive methods or used other methods yourself to look at?
From a Western blotting point of view, I think S6 kinase is the best. If the most sensitive by far is cell size, though. So you can pick up, if a Coulter counter measures cell size, you can pick up sub-picomolar amounts of mTOR inhibitors because you're counting tens of thousands of cells, and so you have lots of statistical power to see that. So you can detect amounts that you could never see on a Western. But that's kind of a pain in the butt way of doing it. So.
Other questions?
All right.
If there's no further questions, we're just left to thank you again.